From Microbe TV, this is Immune, episode number 93, recorded on May 20, 2025. I'm Vincent Racaniello, and you're listening to the podcast about the body's defenders against disease. Joining me today from Ithaca, New York, Cindy Leifer.
Hi, Vincent. How are you?
Every time I say Ithaca, it reminds me of my youth. Oh my gosh. My youth. Remember
your good
that?
Old days.
The good old days at Ithaca at Cornell. Oh my gosh.
What do you remember most from being here? Cold winter?
Yeah, cold winter. Because you're not there in the summer, basically.
Yeah, we love it during the summer. It's absolutely spectacular here,
Yeah.
and all the students are gone. It's great.
Big campus, a lot of walking.
It is a lot. And hills. A lot of hills.
Yeah. And also, there's some nice parks around there. Like, what is it, Taganik?
Buttermilk. Yep. There's
Buttermilk.
If anybody
nice.
ever wants to visit, it's spectacularly beautiful here. You should come.
But I had a rough time. I didn't know what I wanted to do, and everything was hard. So I come out of high school as the salutatorian number 2, right?
Yep.
Straight A's, and all of a sudden I can't figure anything out at Cornell.
It's a pressure cooker environment. A lot of the elite schools are like that. You know, you're the top of your class, and then you go into a classroom where everyone's the top of their class, and not everyone can be the top of the class anymore. And it's a wake up call for a lot of students. You get a little shock. Also,
the professors didn't really care. I mean, they just blather away. I didn't have a lot of good professors, frankly.
Oh, that's unfortunate. I think
but
there are
the science professors, I had Jerry Fink, who was there when I was there, and since went on to be director of the Whitehead, and a couple of other people who inspired me. I mean, as a biology major, and they inspired me to keep going in that. But
did you do research while you were an undergrad?
No, I didn't.
No?
I wasn't connected at all. I just took classes, and some of them were really boring, like zoology was so boring. She didn't make it interesting at all, right? So I'm reading the Zen of Motorcycle Maintenance, right? Did you ever read that book?
No, I have not, but I've heard
Zen
a lot about
and
it.
the Art of Motorcycle Maintenance. It's about a guy who was a college professor, and he said at small colleges, at teaching colleges, you just teach and teach and teach till you get boring, and the students don't understand why you're boring. And I think that's what happened. You just teach and teach every year the same thing, and right? So I think I saw a lot of that. But I learned something, and, here I am. But I say that I was really lost. I didn't know. Because my dad wanted me to be a doctor, and I didn't want to be a doctor. I was not interested. Mm. So I had a real tug. And so I ended up graduating, not getting in anywhere, thank goodness. So I worked in a microbiology lab for a year, and that's where I got interested in virology. So worked out okay.
What was the microbiology lab working on?
It was a company. And it's very funny. I think I can say this on this podcast. So this company made condoms, spermicides, and trichomonas treatment creams, right? Huh. All kind of sexually related things. And so I had to test them for sterility and activity. So there used to be these condoms made out of intestines. God. And they were from lamb, and they would package them. And of course, they're full of bacteria, but they would package them with some antimicrobial. So I would have to take the little capsules and break them open and test them for sterility. They would leave them for 2 weeks in the antimicrobial, and then I would test them for sterility. And they also made latex condoms. It was this big room and with desks and ladies sitting at the desk with these golden phalluses, and they would take dildos. They would take each condom and roll it on and blow it up with air to see if there are any leaks. And then they would throw it in a box. And then there was another room where they made it. There are these dildos on on a conveyor belt, dipping into latex and coming out coated with this thin coating, which would then dry out. Oh my God. I also did, so they had spermicidal creams and I did activity for that. So every week we would get a donor to give sperm. This turned out to be my boss. He would just do it every week. And so I would mix the dilutions and watch the sperm die, And the same, we had trichomonas cultures and I would do that. So I did, I learned how to do some things that I also realized I didn't know anything. So I said, I got to go back to school. And so
is that the part that prompted you to go to school? Or was there something about what you were doing hands-on every day that got you interested in the research?
I really liked doing lab work. I hadn't done much, right, in my career, but then I went there and they let me do things, right? They trusted me.
Yeah.
I remember there was this one thing they would sell little vials of slants where the agar is solidified on a slant. So and then it would go to a physician's office and they could do cultures.
Yep.
And so we would mix up this huge bottle of agar and then it was pumped through this thing that had like 6 eggs, and you'd fill 6 vials at a time, and you'd have to go. You had to have a really steady hand. You didn't want to spray it outside the vial, and they let me do that, and I was really good at it. I'd love to focus to do it, without spilling anything. And then when that whole thing is empty, you got all these thousands of vials. You incubate them on a slant, and So I liked having that responsibility, and nobody looking over my shoulder. So. That's it. Oh, I like doing this. And so that's one of the reasons I said I have to go back to school and get more education. So that's what I did. So
there you go. You can look at sperm dying as fresh out of graduate school or fresh out of
too.
undergrad, and then you can become a virology's world professor.
Yeah. Yeah, it worked. Well, professor. So I was applying to master's programs, and then one night I went to the house. My roommate at Cornell was Ed Kilbourne. And his father, Ed Kilbourne, was chair of microbiology at Mount Sinai, a very famous guy. And so Ed Sr. came. I said, Vinny, what are you going to do with your life? I said, well, I want to be a virologist. I'm applying to master's programs. He said, screw that. Just get a PhD. To hell with masters. Come to Mount Sinai. So this was August. So the next week I went for an interview. I got in and I went. And it worked,
Very cool.
August is very late, as you know, the application process. But
in August, students are applying now to get admitted for the following year.
I know. So, but he's the chair; he can do whatever he wants.
We can't do that anymore.
But I went and I had good grades at.
Oh, that's great. Yeah,
Cordell, and I knew how to talk, right? And so I got in, and then worked for Peter Palazzi. We worked for David Baltimore, and I was lucky. I found my way, right? And then many years later, my dad, he visited my lab. He said, I'm glad you found your way.
Oh, that's nice.
So he was happy, right? Because, what he did, he was a surgeon, a chest surgeon. I didn't want to do that. I wasn't interested. But I didn't know what I was interested in. But I think you have to find your own way, You just need to explore different things. And that
I need you to talk to my kids because they're both like graduating high school and still in college, and they're trying to find their way. Well, you have to experiment, and
coming out of college, you may not be ready. Like I wasn't ready. I worked and I found it, and my parents let me live at home and supported me, so that was very nice, and I had no problem with that. So I remember going to Cornell. No, I remember going to graduate school. So I lived at home for a year, and then I go into graduate school. My grandmother says, you can always come back if you want. I never did. Not to live. She said, if you want, you can live here. But no, I never did. That was it. Yeah. So I consider myself very lucky. But Ithaca was a tough time for me. But I graduated and had a reasonably good GPA,
Yep.
So, and in the end, I loved organic. I just think it was so cool. Had some great biology courses. I had a great microbial genetics course up in Stocking Hall.
Yep. Still there. Renovated.
And it was at night. Oh my gosh, it was at night in this big lecture hall. And it was so hard to stay awake after dinner, And he would just read his notes.
Oh, that's not good.
He gave you a big sheath of all his notes at the beginning, and he just single space typed, and he would just read them. Stanley
I'm surprised you
Cohen.
went to class.
I actually liked the material. It was phages and genetics and that sort of thing is really good. So I had a good, and Jerry Fink was a great teacher, actually. He was really good. The Orgo teacher was really good. I got it. I understood it. My first exam in Orgo, I did like a 20 out of 100. And then at the end, I was acing it. I got it. I was happy. Anyway, sorry. We could edit all that out.
We digress. I think it's good. You should leave it in. It's very interesting. I'm sure that listeners don't know a whole lot about that history of you.
Or anyone. Most of us don't talk about our history, but I think we should be. It is instructive. I always tell students, look, just don't be afraid to fail, not in grades, but trying something, it doesn't work, move on to something else, give yourself some time, but just find what you're really interested in. So I got interested in microbiology, right? Just kind of accidentally. I did read this book called Fever, right?
Mm-hmm. Yep.
I have it here.
Right behind you. Wow.
I have this shelf for this purpose. So this is Fever.
Yep.
This is from 1970, this book. And it's all yellowed, the pages are yellow. But this is the story of Lassa virus discovery. It was the first emerging infection. I read this and I go, oh my gosh, I didn't know viruses could do this. I have to study viruses. So that was really the catalyst.
And then I said, I want to study how the immune system reacts to the virus.
Yeah. So you got to find your passion. And to do that, you have to be open-minded. You cannot be closed. You cannot be critical. You can't be judgmental. You just have to look at things and be open-minded. So, and today we have 2 open-minded papers for you. we are missing Steph.
We're missing the better half of immune.
But we will go ahead.
So
we have one immunologist here, and so I'm a virologist who I like immunology very much. Yeah. Because it's amazing. And I'm reading these papers and I'm like, it is so complicated. I'm amazed it works with all these cells.
Every day I wonder how we are alive.
Because a lot of things have to happen. But that's evolution. Evolution builds good stuff. Yeah. Not always. Sometimes it breaks, but
well, yeah. I mean, if it doesn't work good enough, then you die of infection. And if it overworks, then you get autoimmune disease and chronic inflammation. So understanding how that fine balance is made is what I think is so fascinating about immunology.
So I have a snippet, which is in Nature Communications. It's called T-cell memory response to MPXV infection exhibits greater effector function and migratory potential compared to MVA-BN vaccination. So MPXV is, of course, monkeypox virus, and MVA-BN is modified vaccinia Ankara, which is a vector carrying antigens related. Actually, it's a smallpox antigen. It's smallpox vaccine that works for MPX. So MPX-V is monkeypox virus, and then the disease is called MPox. Okay. You have 2 different things. And this is coming from, it's at the end of the paper. Looks like a lot of people in the UK. I don't want to scroll all the way to the end. But the first author is Jili Chen, and the last is Tao Dong. So, all right, well, I'll scroll to the end just to be fair to the authors. Here we go. I don't like that they do this. There's a lot of...
Yeah, they did put it in weird place.
There's tons of affiliations, but mostly in the UK. I see. So as you know, lately we've seen a lot of MPox disease caused by monkeypox infections. And in fact, since smallpox vaccination was stopped in 1979, there have been more and more monkeypox virus infections because there was some protection conferred by the smallpox.
I didn't realize that they were that closely related. Yeah,
they are. In fact, the vaccine we use against monkeypox virus is a smallpox vaccine. It provides because we don't have an MPX vaccine itself.
I guess those viruses are similar too, because if you think about the smallpox vaccine came about because of cowpox. So cowpox and monkeypox and
so we have
variola.
These are the pox viruses, right? We have smallpox or variola virus. We have cowpox virus. We have vaccinia virus, which most people use in the lab as a pox virus, and then monkeypox virus. They're all very closely related. And monkeypox virus, there are two clades that have different geographic ranges and different case fatality rates. It's really interesting. Clade 1, or in Central Africa, they have a case fatality rate of 5 to 8%.
I didn't realize
which is really
that they
high.
were different, and that is really high.
And Clade 2 comes from West Africa with a case fatality of less than 0.2%. And the current global epidemic, actually from 2022 to 24, was caused by Clade 2. 95,000 cases, 178 deaths. But these continue to transmit in many countries, and there are cases in the U.S. But more recently, there's been a Clade 1 outbreak in DRC, and that has killed over 1,000 people. Okay, it's quite lethal.
That's scary.
So this vaccine, MVA, so Modified Vaccinia Ankara, BN is Bavarian Nordic. It's the company that makes it. It is a third-generation smallpox vaccine. So it was made, the original smallpox vaccine had a lot of side effects. So they made second and third generation, and this is one of them where they use a vaccinia virus, Ankara is the strain name, to vector the antigens. And this has some effect protecting against monkeypox disease, right? And this vaccine induces neutralizing antibodies. And they know that the antibody response is pretty weak, but there's a really good T-cell response to the vaccine.
I was wondering, because with antibody, really good antibody titers, we think about that with association to mucosal infections, like respiratory infections. But the skin, how I should probably know more about this, and I'll go look it up. But I don't know how effectively the antibodies are secreted at the skin.
Yeah.
I don't think very well. I mean, they're going to be right underneath the skin, or if you damage the skin or have breaks, then you're going to get exposed to antibody. But I'm not sure. I would think T cells would be more helpful. So I don't know how important antibodies are going to be for protecting skin versus respiratory, but I think T cells are going to be pretty important. So it's good that they're looking at T cells in this paper. Yeah,
so that's what they're looking at in this paper. And they use vaccinia virus because there's 84% nucleotide sequence identity between monkeypox virus and vaccinia virus. And a lot of the T-cell epitopes are conserved between the two viruses. So why? Because it's kind of dangerous to work with monkeypox virus. I don't actually know what the containment level. Let's look.
Well, are there treatments?
Really, the antivirals don't work.
Right.
There were some of the smallpox antivirus, but they don't work.
So if there's no treatment, it's got to be at least BSL-3, right?
Probably, but I'm not sure. I couldn't find it. But anyway, so they develop, they look at T-cell responses, basically. They have a cohort of 13 people in the UK. These people recovered from MPX in August 2022. And it was confirmed that they were positive, and some of them had skin lesions, et cetera. And then they have 10 controls that didn't get infected. So that's the convalescent cohort, right? They recovered. Then there's a vaccine cohort. They have, and they're all men, by the way, in both cohorts. 20 males after vaccination with MVA-BN. And none of them had ever been infected, although 2 of them had been vaccinated for smallpox before 1980, which is when we stopped.
Yep.
And in this study, they were seropositive for...
That's amazing.
Isn't that cool? It lasts so long.
Yeah.
It's very good. I mean, this is vaccination, right? Where they scarify the vaccine into your skin. So amazing. Okay, so they isolate PBMCs from these individuals, and they look at interferon gamma production. And they stimulate the PBMCs with the vaccinia virus strain. So they don't want to stimulate them with MPox. So they use vaccinia, but that should be fine because they're quite cross-reactive. And so you can see the T cells, the ones that react against vaccinia are making interferon. You can see it on the assay, which is called an Ellis spot. So it's kind of an ELISA in the spot format, right?
Yeah. So you basically make a plate, and when you plate your cells, and then when you go to analyze them, you put a little gel over top of them so they stay in place.
Right.
And then you're able to see where they've made the antibodies. And so each cell is producing antibodies, and they stay relatively locally so that you can, the plate is also coated with antibodies against the target antigen. So then each cell, when they're producing the antibodies, they make it locally, and then you can detect the spot when you go to develop it.
So they find that the people who recovered made a good antiviral T cell response compared to healthy controls, right? And they also looked at responses to influenza virus, Epstein-Barr virus, and human cytomegalovirus, and I guess as controls, and they were comparable in the two groups. Okay,
which it should be, right? Because they all should have been exposed to those.
For sure.
Yep.
So then they do an assay that Cindy and I were talking about, an AIM assay, activation-induced markers. So these are looking for markers that are present when T cells are activated, right?
Yeah. So the way you usually assay T cells, there's a couple different ways. One is that Ellie spot. So that will tell you the number of cells that are making cytokine. And the intensity of the spot may be sort of a little bit of quantitative, but the quantitative aspect is how many cells are there that are responding specifically. ELISA will tell you the total amount of cytokine, but can't tell you how many cytokines are making it. But now this assay sort of is a single cell assay of level of response, because you can look at surface markers that come up after activation, and the level roughly corresponds to the level of activation. So instead of just using one marker like CD69 that some people use, they use multiple markers, and that multiplexing allows them to really accurately identify the cells that are responding specifically, especially if you incubate with a particular virus or peptides. You can see the specific T cells responding. And
you look for specific markers, right?
Yeah, there's different combinations. I was writing them down. I think we were talking about that right before we started. So oftentimes, for CD8 T cells specifically, they're often using CD69 in combination with another marker that comes up, and that can be 4-1BB, CD40 ligand, PD-L1. They're often going up, and CD4s similarly have sets of dual markers that they look for upregulation. And then if they upregulate both, they classify that as a responding cell, and then it's positive by the AIM2 assay.
So they do this to look at both CD8 and CD4, reactive 4 and 8 cells. And they find the proportion of CD4, CD8 reactive T cells varies between participants, but all of the individuals show some kind of CD4 and CD8 T cell response.
Yep. It was weird. There was one that had only CD4 and one that had only CD8. I don't know if that's a level of detection or something quirky about those individuals. I thought that was weird.
It's true. It's true, yes.
But they all responded at least some. Yep. So that's good. They had T cells specific.
Then they want to know what are the epitopes that are being recognized, right, by the T cells. So they make peptides, 232 CD4 peptides and 273 CD8 epitopes. So you can go and look at databases to figure this out, right? You don't have to reinvent it yourself.
Nope.
And these are quite conserved between vaccinia and MPox virus, right? There's CD8 epitopes, 78% are conserved, and the CD4, 72%. So they feel okay using vaccinia in these experiments. And then they add the peptides to the PBMCs, and then they measure Ellis spot responses, interferon gamma production. And they say overall there was a greater CD8 response to proteins made early in infection. So when DNA viruses infect cells, they're early, middle, and late phases for various reasons. And they find the early proteins are most of the T cell epitopes compared to the intermediate and the late ones.
I don't know why that would be, but it is a nice coincidence because if you'd like to be able to recognize infected cells early, rather than having to wait till after they're already making copies.
Yeah, I think that makes sense.
Yeah.
But they don't see much of a CD4 response. Only 2 individuals had a CD4. The others much lower than this. Well, those 2 individuals, it was lower than the CD8, and the others didn't make any. So that's kind of interesting, right?
I don't know what that means. Maybe it's just that in the blood, there's less access to those CD4 T cells that of interest. Maybe they're tissue resident cells or surveying lymph nodes and less likely to find them there. I don't know.
Yep. So then they focus on the early epitopes. They make overlapping peptides to the 5 early proteins of the virus, the ones that are expressed first when the virus gets into the cells. And they again do the Ellis spot assay, and they see various responses for the different peptides. 7 of 12 recognized at least one of the antigens, and there were a couple that had the greatest immunogenicity. So they say, well, maybe these peptides could be part of a future vaccine, right? To add T cell reactivity to the vaccine. Let's see. What about function? So are these responses functional? And first they find that there's a very common MHC1 allele, and it's HLA-A-A02, okay? And so obviously many of their participants are positive for this. And so they take PBMCs from them, and they identify peptides that are reactive with that MHC. Because remember, the MHC presents the peptide to the T cell receptor, right?
Yep.
So they have a whole list of peptides that react with HLA-A2, and some of them are conserved between vaccinia and MPox virus. So they can find immunodominant epitopes that bind this MHC1, which is very common. So that's good. That way you don't use an MHC that's 2% of the people or something. It's not useful, right? Then they add these peptides that hit this particular MHC1, and they add that to their PBMCs. Actually, they use T cells that are transformed with EBV. They're B cell lines transformed with EBV. So they co-culture the T cells with these with vaccinia virus infected. So no more peptides. We have a real bona fide infection here, and they show that all of the T cell lines respond to these vaccinia virus infected B cells, and they make cytokines, and some of them make multiple cytokines, which they call polyfunctionality, right?
Mm-hmm.
And they also show that these so these are CD8 epitope-specific T cell lines, right?
Yep. They
can kill the vaccinia virus infected target cells. So the fact that they're B cells is just because they're transformed by EBV and they grow. They're immortal. Yep. People use these all the time, right? It has nothing to do with what they're looking at.
No, it just does a target.
Yeah. Yeah. And so these can kill vaccinia virus infected cells. So these peptides that they've identified are potentially functional, right? That's good. So now that's all convalescent so far.
Right.
now we go into the vaccinated people.
Right, because it's a good question. You want to know if you vaccinate, can you generate a similar response as you get with the natural infection? That's always a question that people have, right?
Yeah. So they do the ELISpot again, just to, as you said, numbers of T cells, right? Yep. And they're similar between convalescent and vaccinated individual. But then they drill a little deeper and look at phenotypic analysis of these cells. And they use, again, these HLA-A2 positive donors. And they have, so they use tetramer staining, which a tetramer is a HLA molecule, basically 4 of them together. Is that right?
Yeah. So tetramer means 4, right? So it's got 4 MHC molecules that are folded properly and then bound to peptide, and they have a fluorescent tag. So basically what you can do is capture those T cells that have specificity for that. And so it can give you an indication of what is the percentage of specific T cells that can bind to that MHC peptide complex.
So they use these to stain the CD8 T cells from both groups of people. And they find that the T cells from convalescents, the frequency of these are much higher in convalescents than in vaccinated individuals, right?
Yep.
But no difference in memory phenotype of these tetramer positive T cells between convalescent individuals and after vaccination. So I guess that makes sense, right? Because maybe the initial difference is the acute phase of the infection, and the memory phase, they're more similar. Yeah. I don't know. And then, of course, they do RNA-seq.
Yeah. Every immunology paper has to have RNA-seq in it these days.
So they sequence the transcriptome of these T cells. This is from convalescent individuals. And they see an increase in certain markers. And you can tell me what these mean, because they don't actually tell us the FOS, DNA, JA1, NR4, CD69, GLIN-Y, and GZMA. I don't know.
They did not do a good job of explaining why we should care about these. Yeah. I just believe they're mostly just activation markers.
Okay.
Granzyme A, and the
the point here is that these are more are increased in the convalescent compared to vaccinated individuals.
Yep.
Right, whatever these markers mean, they're higher.
But it's important to note that this is not very many cells. So when you do single cell RNA-seq, most people these days are in the tens of thousands to 100,000 cells. These are 400 and 160. It's not a lot of cells.
Yeah.
It's a couple hundred cells to 1,000 cells at best. So I worry a little bit about distinguishing what you can tell difference with those low cell numbers, VIC single cell RNA-seq. I will say that the violin plots, so those neat little, they almost look like a candlestick. Those are violin plots. And it is statistically significantly different for the markers that you just mentioned between the convalescent and the vaccinated. So there is a difference for sure. But what that difference means at the protein level and the functional level, those are two leaps that we make from single cell RNA-seq data that sometimes are correct and validated and sometimes not.
All right, then their last... Then they look at T cell receptors, and they see expansion of T cell receptor alpha and beta just in the convalescent samples.
Yeah, the only thing I thought that was interesting about this is that there were much more narrow usage of TCR alpha and TCR beta for the convalescent. So there are these, I don't know what they're called, they're like a disc, and they connect. If it has this alpha and this beta, and if the band across is really thick, it means that most of them use this alpha and this beta. So to me, the only thing I took away from this is in the convalescent, their TCR choice is much more narrow. When you vaccinate, there's many more TCR alphas and TCR betas that are made, and they pair more. I don't I don't know what that means. They talk about what are called public clones, which are things that, a TCR that you find in more than one individual.
Yeah.
So it might suggest that the infection drives expansion of specific TCRs more than the vaccination.
Okay. Then the last set of experiments, they are, again, using RNA-seq to look at expression and comparing these CD8 T cells after infection and vaccination. And they're also going to compare it to SARS-CoV-2 specific CD8 cells. And what they find is, I'm going to summarize it for you in the interest of time. Basically, convalescent MPX-specific memory T cells in convalescent are more cytotoxic than in after vaccination. They're more activated, and they produce or they express transcripts encoding migratory molecules. So, right, the rash is in the skin, so the T cells may have to migrate there. So maybe these memory cells are ready to go upon reinfection. They also find that the response, the cytokine and chemokine expression of these MPX T cells are lower than that of SARS-CoV-2 T cells. But I don't know what that means. It could be methodology, right? Who knows? But I think it's interesting that they express migratory markers, right? That's probably the most interesting thing.
Yep. But we have to remember there are also memory, skin resident, tissue resident memory T cells too, that you're not surveying here. You're surveying the ones that you can pull out of the blood. So, it's true.
I guess a fraction of those may migrate to the skin, right? Yeah,
definitely. After reactivation, sure.
So the vaccine and the infection makes T cells. The infection is better, makes better T cells, we'll say. It's probably not surprising because you have more epitopes in it, I guess. Maybe you could use this to improve the vaccine, which the vaccine also induces T cells, but maybe you could make it better. And they say, the vaccine is administered like kind of intradermally, right? But just by scraping. And so they say that intradermal vaccination may better prime T cells at the infection site, as you said. And so maybe you have to look there. You'd have to do that in an animal model, right?
Very likely. Yeah. I mean, I suppose you could biopsy human skin and look for those cells.
So there you go. I think it's important to look at T cells, and early in the COVID pandemic, everybody's looking at antibodies because it's easier, right?
It is much easier to analyze antibodies.
Yeah. But then we get going. But all the vaccine recommendations are based on neutralizing antibodies, not T cells.
That's right.
And I was talking to a guy from CDC years ago, and he said, you'll never see a T cell based recommendation.
No.
Forgot why. Okay.
Because you just can't measure it, so it's harder to make a suggestion based on it. Yeah. Well, that was interesting. So I picked something completely different. And actually, it's kind of funny because before you picked that snippet, you also picked this paper and you sent it out. I'm like, wait, Vincent, that's the one I picked. You can't do the same one.
We picked the same paper, yeah.
So great minds think alike. So it's a pretty cool paper, I will say. So the title of the paper that I picked, and it is published in Cell in April of this year, is Macrophages Direct Location-Dependent Recall of B cell memory to vaccination. And I thought this was really cool because at the end, they really talk about why you might or might not want to vaccinate in the same arm. And that's always been something that has been a question. And here they really provide some mechanism for that. And they do a study. So we'll get to that. But there are, don't want to shortchange anyone, two co-first authors, Rama Denny and Alexandra Carey-Hope. And there are 4 last authors. So this was a big study. And if I get this right, it's Anthony Keller, Vanessa Venturi, Si Mingling Munier, and Tri Gang Fan. And all of the authors, as far as I can tell, are located in Sydney, Australia. So it's a study from down under. And the highlights, if you don't want to listen to everything, is basically there is definitely a difference between the draining lymph node and the non-draining lymph node. So what does that mean? It means if you immunize in your right arm, your right axillary lymph node is your draining lymph node. So anything you inject in your right arm will drain to that right lymph node. And then the non-draining lymph node is in the other arm, because that's not where, the skin has lymph. The lymph collects the antigens and then it puts them through afferent lymphatics and drains them into the lymph node. And we'll talk a little bit about lymph node structure. But the idea here is then if you go and boost in the same arm, then that same antigen is going to drain to that same lymph node where there's probably memory B cells, memory T cells, follicular dendritic cells, and etc. Whereas if you immunize in the other arm, those are not there. So are you generating a new response, or are you generating this, a boosted memory response? So that has been a question. And the cool thing is they are able to show that there is different trafficking of memory B cells in these draining and non-draining lymph node. So there are memory B cells to the antigen in both lymph nodes, but they traffic differently, and that changes their function. So then, the question becomes, should you boost in which arm, which we'll get to. So to introduce this, we know that vaccines work because they can induce both memory B cells and long-lived plasma cells. And we talked about long-lived plasma cells in Immune 86 when we talked about where did all the antibodies go. But long-lived plasma cells are important because they produce antibody right away. And they provide a really nice first layer of defense against reinfection because they will be boosted and they will start producing that antibody, and that antibody will circulate, and that protects. But they're fixed in their specificity and affinity, and they also wane over time. So you lose kind of those long-lived plasma cells, but that's where then you have B cells. So memory B cells can rapidly differentiate into both short-lived plasma cells, or, and this is the important part, they can enter or re-enter germinal centers. And germinal centers are really important parts of lymph nodes where a couple of important processes happen. What happens there is that you diversify the B cell receptor, also the antibody. What does that mean? That means that you take the sequence that the B cell makes for their antibody and you start messing with it, and you make what's called somatic hypermutations. So you make mutations and then you test those to see if they're better or worse or no change. And that's called affinity maturation. And what that does is allow you to refine the specificity of the antibody over time. So you get better and higher quality antibodies from the B memory cells. So this fate of this B memory cell is really important. Does it go down to these plasma cells? Does it enter germinal centers? Does it make the antibody response better? So that's why looking at where and how these B memory cells are moving and functioning and what they're doing is really important. And so there's been a long-standing discussion, should you boost the same arm or different arms? Where are the memory B cells? How are they functioning? And we actually talked about this also with Gabriel Victoria when he was on the show in Immune 52. So we can go back and look at that one too. But generally, the other important thing we need to know about this paper is how do antigens get into the lymph node? So textbooks and what everybody teaches usually is their dendritic cells are in the tissues. When you get a vaccine, those local dendritic cells will take up the antigen. They also get stimulated by an adjuvant. They go through a maturation process. They migrate to the lymph node, and then they activate T cell responses. But there's also antigen that comes just straight into the lymph node. So when you inject something in the tissue, antigens can drain directly to the lymph node, and there they can be taken up by different cells that we'll get into and brought into the lymph node and allow B cells to respond. They can also bind things like antibodies, if you already have them in the tissues, or complement, and then those bound things can also come in. So the structure of a lymph node is afferent lymphatics come in and they drain into something called the subcapsular sinus. So the capsule is just the thing that surrounds the lymph node. And then right under that is an opening. And so the antigens and the cells and whatever that's coming in the lymph node from the lymphatics, from the tissues, drain into this subcapsular sinus. Now there are cells, which I love, macrophages, and there's a specific type of macrophage that lines that subcapsular sinus. That's called a subcapsular sinus macrophage. Their goal is just to basically grab onto anything coming in to the lymph node through the lymph, either directly phagocytosing it, binding to the complement through complement receptors, or binding to the antibodies through FC receptors, bringing it in and putting it into the main part of the lymph node, which right under that subcapsular sinus is called a cortex. And that's where a lot of the B cells hang out. So if you bring in antigens, you can give them right away to the B cells. So if you have memory B cells, that's one good place for a memory B cell to hang out, because those macrophages are going to grab onto the antigens, bring them in and hand them to the B cells. And the B cells can then say, hey, is this something I recognize, and decide whether to respond. The other place the antigens are, which is the traditional way we think about it, is within follicles or germinal centers where B cells have already done their proliferation and they can do their activation part. They're called follicular dendritic cells. And these are interesting cells because we call them the buffet. So they hold out all of the antigens on their dendrites for B cells to then come and sample. And they can hold on to antigens for a very long time, years, we think. So those are different ways that antigens can get into the lymph node. And everybody focuses on these follicular dendritic cells and being able to activate memory B cells. But this idea of when you inject antigen in, if you get exposed to something, a lot of it's going to drain right into the lymph node and be picked up by these subcapsular sinus macrophages. So basically, this paper is asking, does that play any role? So are these subcapsular sinus macrophages important? Are they important for B memory development? And how does that work?
So, Cindy, when you get an injection in a deltoid, say, does most of the antigen go to the axillary lymph node, or does some circulate?
I don't know the exact breakdown, but it will be some and some.
Yeah.
So some will stay local and be taken up by cells in the region. Some will be taken up by the dendritic cells, and those dendritic cells will move. And some will just be flushed from the lymph that's bathing the tissues and then drained into the lymph node. And I don't know the exact breakdown of what percentage comes in which way.
Because if you get a second shot in the other arm, you do make a response.
You do.
Right.
Yep. And there's memory B cells that you will find in that non-draining lymph node from the first injection. And they look at that in this paper. Okay. Because those memory B cells are going to circulate throughout the body, and they'll reside in various different locations, some of which is in the non-draining lymph node. You still have more in the original location, but there are some in the other arm. And the important thing is they move differently. And they're able to do this because they use mouse models where they can control the antigen, they can control the specificity of the B cell, they can control the specificity of the T cell. So they can look and they can identify those B memory cells specific for that antigen in the draining and non-draining lymph node, because that's a really cool part of the study. So they use a lot of really cool tools and ways to look at this. So the first question they ask really is, where are those memory B cells, and are they similar in the draining lymph node and the non-draining lymph node? So to do this, they have a very defined system. One could say it's artificial, but it gives them really a lot of power to look at the memory B cells and to control the whole system. So the way they do this is they take what's called SWHEL B cells. So these are B cells that are it's a germline transgenic mouse, in which all of the B cells are specific for hen egg lysozyme. So you know what the antigen specificity is of the B cell receptor. So they can take the B cells from those mice. And then they also have other mice that are what are called OT2 mice. So these are CD4 T cells that are specific for ovalbumin. Now they can mix those and put them into another mouse, into a black 6 mouse. And then the next day they can inject with an antigen that's called HEL ova. So it both has the antigen that the B cell recognizes and ova that the T cell recognizes. Could you do this with T cells specific for HEL? Sure. But it actually is very cool that they did it this way, because in a later experiment, they are able to manipulate this, which is kind of cool. So they have this HEL ova, and what that does is activate the B cell, activate the T cell. You get an immune response. The T cells provide the help to the B cells. The B cells make their antibodies. They do their affinity, and then they calm down, they rest, and they become memory B cells. So they rest the mice for 4 weeks. Now they have memory B cells that are specific for HEL. And now they can ask a bunch of questions. So the first thing they do is they ask, where are those B cells? And I won't go into all the markers and everything, but basically they use whole mount microscopy and staining. And what they see is that in the draining lymph node, they are seeing that the memory B cells go into patrol in 2 places. They patrol deep in the follicle, which is where you would expect them to be. And they also patrol basically right along the subcapsular sinus macrophages. So they're basically looking to see if those subcapsular sinus macrophages are bringing anything in that looks interesting to them. And the difference between that draining lymph node and the non-draining lymph node, so same specificity B cells, but they're just in the other lymph node, is the B cells in the non-draining lymph node stay deep, and they don't ever go up really and survey these subcapsular sinus macrophages. So there's a fundamental difference in where these B cells are, how they traffic. So they have really cool 2-photon in vivo, or not in vivo, but like of a lymph node microscopy of the cells moving around in living lymph nodes. And they can show you the tracks and the speed and how far they move and all of it. They make all these different measurements. But the bottom line is that in the draining lymph node, these B memory cells really skirt along and interact with these subcapsular sinus macrophages looking for antigens. So that's really cool. So why
is that like new? Because I always thought they went into the germinal centers.
So as far as I knew, and what most people study, is that they're in the germinal center.
Yeah.
And I think it's because they haven't really been able to look at this fine localization and trafficking. So when they collect and they're looking at memory B cells, they're basically just looking at all the bulk memory B cells. So they can't tell the sublocalization of where they are in the lymph node. So that's the true advance, the first advance that this paper shows, is really they looked at where are they. And so the location seems to be really important. So if they're right, and this patrolling thing is important, if they take away those subcapsular sinus macrophages, they would hypothesize that it's going to change the localization of those B memory cells. So they do that by adding, so subcapsular sinus macrophages depend on something called CSF1 for their development and maintenance. So if they block that by treating the mice with anti-CSF receptor 1 antibodies, they basically deplete them. And they have a beautiful supplementary figure where they show the subcapsular sinus macrophages are all lit up in the wild type, and then when they treat with the CSF1 receptor, they're just gone, pretty much. So then the question is, does that have any effect? What happens to the B cells? So the one caveat to this experiment is that there's another population of macrophages in the lymph node that is also dependent on CSF1 receptor. So you do lose those two. They argue that that's not quite as important because those are medullary. So if you think about the structure of the lymph node again, if we go back, we said the afferent lymphatics are coming in, they're draining into the subcapsular sinus. The subcapsular sinus macrophages move things across, and then they drain through the lymph node down to the medullary cavity, and then things drain out from the medullary cavity into the efferent lymphatics and out of the lymph node. And then they can get back in the blood through the thoracic duct. But basically, they come in from the top and they drain down to the medullary cavity and out. There are also macrophages that line that medullary cavity, and their job is to make sure they grab onto any leftover antigen so it doesn't get back out into the efferent lymphatics and into the blood. So they're also lost. So that's a caveat to this. But they treat with a CSF1 receptor, and they're arguing that these subcapsular sinus macrophages gone. So what happens to the B cells? They change. So they actually redistribute their localization and their trafficking in their scanning, they look just like the B memory cells in the non-draining lymph node now. So there's definitely something about the initial immunization that changes somehow these subcapsular sinus macrophages that allows the B cells in that lymph node, not in the other one, to be scanning and looking for that rapid bringing across of those antigens when there's a new infection. So that's pretty cool. Yeah. I think. But the question is, does it matter, right? They behave differently in these two, but does it really make any difference? So the next thing they did was test that. So they're going to test whether if you boost in the same arm or you boost in the different arm, what happens. And so what's called ipsilateral and contralateral. So ipsilateral means same arm, same lymph node, draining lymph node is activated again. Contralateral is the opposite arm, non-draining lymph node. And they boost with the same antigen, the HEL ova. And they look, at 5 weeks past, and then they look for what's happening in the draining lymph node and the non-draining lymph node. So draining lymph node first, they see clusters of reactivated B memory cells at day 5, and they see secondary germinal centers. That means those B memory cells are going back into germinal centers and doing their thing with somatic hypermutation. And generally, they see just more germinal centers overall. So that would say boosting the same arm, better B memory response. Okay. So if you boost in the other one, there are clusters of B cells, B memory cells, and they seem to be confined to what are called subcapsular proliferative foci. And they also differentiate more into plasma cells in the medullary cords. So if you're hitting the non-draining lymph node, they go rapidly to the medullary cords and start pumping out antibody. Whereas if you go with the same arm, what they do is they go back and they try to refine to make better antibodies. So they go more to germinal center and more to that path. So what does that mean? That just means that if you boost in the same arm, and this is mice right now, if you boost in the same arm, you're more likely to go towards refining the antibody specificity. If you boost in the other arm, you're more likely to just start taking those specificities you already have and just making more antibody quickly. So that outcome seems to be different depending on how you immunize. And they have numbers, and there's 10 times more antigen-specific germinal centers in the draining lymph node than in the non-draining lymph node. They look at different time points, like 15 weeks, and basically it's similar, although less. And they conclude from this that the germinal center response peaks in the first week after boosting, and more so in the draining lymph node than the non-draining lymph node. So I thought that's interesting. It peaks at 1 week. That's really fast for that to happen. And that it does seem to drive these B memory cells down different paths depending on if you inject in the same or different arm. So the next thing they do, I thought was there's 2 very cool tricks that they do in this paper, and I think they're worth mentioning. The first is they say, okay, what about affinity? What does affinity have to do with this? So the cool tool that they can use is they have the B cell receptor that's specific for Hennig lysozyme. You can use wild type Hennig lysozyme, or you can use Hennig lysozyme that has 3 mutations, so it binds with much lower affinity. But it still binds. Now you have an opportunity to see whether those B memory cells can refine their specificity and now bind better to the mutated version. So they can detect the difference of the affinity to binding to this three times mutant one. It can be low affinity, or if they mature, they can be high affinity. So you can distinguish this difference by flow.
It's cool. That's a good essay. Yeah,
so that is really neat. So then they can say, okay, well, now if we boost, same or different, what happens? Do we have a better ability to generate now high affinity B cells to that mutant version of the antigen? And the answer is yes. So if they boost on the same side, they get more germinal centers and more high affinity antigen specific B cells to the mutant version. So that fits with what they saw was there's more germinal center response. And then they also did a cool blocking experiment, getting in the weeds a little bit. But if they blocked CD40 ligand before they went and did the boosting, the idea was if there were any persistent germinal centers that were still ongoing, they block them and get rid of them so that they're really looking at triggering of new B memory responses. And what that suggests is that it really is new B memory responses and not persistent ones. This comes into play because if you remember when we do the mRNA vaccine, there's actually quite long persistent germinal centers. So we reboost at 3 weeks, but we have persistent germinal centers that are still ongoing at that time. So that was a really neat experiment, and it suggests that, this germinal center type of response really does play a role in refining the specificities. They also looked at T follicular helper cells, and I thought this experiment was the most clever experiment in this paper. And that is because they were using those antigen-specific T cells, and they took the OVA, OT2 specific cells, and they had the HEL-specific B cells. They can use a different T cell to a different specificity because we've got multiple ones that people have made over time. Therefore, what you can do is generate the original immune response with the OVA-specific T cells and the HEL-specific B cells. Then you have memory B cells and memory T cells, memory T follicular helper cells to help those B cells. The T cells are specific for OVA. Now you can put in a different T cell that recognizes a different antigen. And those are naive T cells. They are not memory T cells. And you can ask, does the fact that you have those memory T cells in the same lymph node matter? Because what they did was take something called a SMARTA T cell. Those are specific for LCMV peptide. So now you have naive LCMV specific T cells in the mouse. You have antigen experienced T follicular helper cells that are specific for OVA and B cells that are specific for HEL. Now you can compare if you immunize with HEL OVA, so you're triggering those old memory T cells, or you're triggering with HEL GP61, which is the peptide that can now activate those new T cells. And you can also ask if you're doing it in the draining lymph node or the non-draining lymph node. So you can ask every single combination. So it got very complicated. But what did they find? They basically found that the new naive T cells were much worse at promoting the B memory response. So, and it really didn't matter whether it was draining lymph node or non-draining lymph node because they were naive cells. So, basically, you want the memory T cells and you want them in the same place. So that's really the bottom line. There was a lot more data in this paper. They basically, the last important thing that they did for the mouse studies was ask, do you really need those subcapsular sinus macrophages for the good response? Because that's really the big question you want to ask. So again, they treat with the anti-CSF1 receptor, and they found that there was an 80-fold reduction in the antigen-specific B cell response in the draining lymph node, not affecting the non-draining lymph node. So what does this mean? This means that when those antigens are draining, those subcapsular sinus macrophages are really important for positioning those B memory cells and delivering the antigen to those B memory cells very early so that they can then go and refine their specificity. And this really happens in the draining lymph node and not very much in the non-draining lymph node, because when they depleted the subcapsular sinus macrophages, it didn't really do much to anything to those memory B cells in the non-draining lymph node.
But why is it different in the non-draining? Right? It's just distance, right?
So distance is one of them, but they did the obligatory single-cell RNA-seq experiment that you see in every immunology paper. And what they conclude from their single cell, and I'm not going to go through all of it. If you love the single cell, please check out the paper. Their conclusion was that there's something fundamentally different about those subcapsular sinus macrophages in the draining lymph node than in the non-draining lymph node. And they did an experiment to test this because what they did was they treated the non-draining lymph node with a different immunization that's not the ova, so that you would get the same kind of response, just not antigen-specific response. And then they ask, was that enough to boost the ova memory B cells? And the answer was yes, to some extent. And what they conclude from that experiment is that the immunization itself drives some change in the subcapsular sinus macrophages that makes them better primed to pool those B cells and make them scan them, so that when you generate a boosting response, those memory B cells more likely will go to germinal centers and generate the refined response.
That's the obvious conclusion, but why? Like, why not the antigen goes to the other one and does the same thing? Right?
Yeah, so I can't answer that, but I can say that if you think about a human who's getting exposed to things all the time, my presumption is most of our lymph nodes probably already have these heightened activated subcapsular sinus macrophages. Because when we're using these mice, we're using super clean mice, and you're injecting in one side and then you're being able to see these differences. And so it could be a cleanliness in the mouse, in your mouse studies type of thing that we're able to visualize, and that it becomes less relevant over time, because once you're exposed to antigens in every which way, now your lymph nodes, maybe that's just the normal way lymph nodes should function. Yeah.
Most of us are picking up antigens
all the time,
systemically, right? To breathing and eating.
Yep.
And so they're distributed, I guess. Right?
Well, each thing. So when you eat, there are mouth draining lymph nodes. When you breathe, there are lung draining lymph nodes. When you get breaks in your skin, like I cut myself putting in a garage door opener on Sunday, there's things that get in your skin, and there's, lymph nodes that drain. There's hundreds of lymph nodes all over your body.
The vaccination is a special situation where we direct it very clearly to one draining lymph node. So, and that shows you what happens when you do that. Yeah.
Yep. We should all have a very good, robust lymph node under our arm. I don't know. Do you always get your immunizations in the same arm, or do you vary them?
I don't remember. I just don't. I remember the last time I got 3 vaccines. So I did 2 in one arm and 1 in the other, just so that one arm wouldn't be so sore. But I don't keep a record. So I should just say always right arm, and that would solve the problem, right?
Are you left-handed?
Yeah. No, I'm right-handed.
Oh, you're right-handed and you always get in the right arm? I always get in the left arm because I'm right-handed. That way I don't have a sore arm that I'm the dominant
but I
arm.
never got, like when I give blood, it's always in my right arm.
Yeah.
I never had any effects, but in terms of these draining lymph nodes now, I'm going to make sure it's the same arm.
Yes.
Well, I don't know if it makes a difference clinically or not. That's the key, right?
That's the big question, right? So the last set of experiments they did was they actually went in human. And I think that's why this is, a more important paper than it otherwise would have been. So they took advantage, as most people did in 2021, where they had opportunities to have individuals who had not had SARS-CoV-2 and who had not yet had immunization. Now, their definition of no previous SARS-CoV-2 infection was they collected serum and tested for IgG antibodies to SARS-CoV-2 using a test that they have, and they tested negative. So we have to, there could be some individuals that actually had it, but their serum antibodies are low, but generally that's a reasonably good way to do it. They had 30 healthy individuals with no previous infection, and these individuals received a vaccination and then a booster, and they receive a booster in either the arm that they got it in before or the other arm. So they were able to test this. And so they controlled for age and gender. So they know that this is going to be due to, which side they got the immunization in. And what they were able to do, which is amazing, is they were able to get fine needle biopsies of both the draining lymph node and the non-draining lymph node.
Wow.
5 to 7 days after the boost, in 18 participants, 9 that got the boost in the draining lymph node side and 9 that got it in the non-draining lymph node side. So you've got draining lymph node and non-draining lymph node from individuals who got reboosted on the draining side or the non-draining side. So you got all the combinations, and they have fine needle biopsies, and they can look at what is happening. Now they can't do the, subcapsular sinus macrophages and the B cell trafficking, but they can look at functionally what's going on with these B cells. So what did they find? They found that if they boost on the draining lymph node side, there's an increase in the number of germinal center B cells and plasma cells. So that makes sense, but there's a nuance. It's because if they do it in the non-draining lymph node side, they see increased serum ELISA of anti-spike IgGs was higher. But that was short-lived. The draining lymph node. If they went in the draining lymph node, they saw serum ELISA went up for 5 days and then waned pretty quickly. So this kind of fits with what people think, right? So you get a boost, antibodies go up, and then they wane. No surprise. If you boost in the non-draining lymph node, and here's the nuance, the response was delayed. So the peak was at 4 weeks. So if you look at 7 days and you compare draining and non-draining, you're going to say draining lymph node, absolutely should always get it on the same side. But if you look 4 weeks later, there's actually no difference. So at least for the serum antibodies, no difference. What about the quality of the antibodies? So are they neutralizing? So and they did their biased experiment here. They looked at 5 to 7 days, the draining lymph node, the boosting on the draining lymph node side had higher levels of antibodies to the ancestral strain and also generated better broadly neutralizing antibodies to Delta Omicron BA1, Omicron BA2, and Omicron BA5 at 5 to 7 days, which we already said we peak higher with the draining lymph node immunization side. So I don't know, but by 4 weeks post-boost, they're basically the same.
I guess if you're in a hurry, you want to go travel right away, you get it in the same arm, right? Right.
If you know you're going to be exposed in the next week, get in the draining lymph node arm. It doesn't matter, then it doesn't matter which side you get it into.
So I guess they could do a clinical study looking at IPS versus con and looking at disease, right? Disease outcomes.
Yeah.
And
they did not do that in this.
They didn't do that, and probably nobody's going to do it. Just do it in the same arm, and
Yeah.
Because it doesn't hurt to do it in the same arm.
Nope.
Interesting.
Yep. They did some mathematical modeling, and they also, interestingly, if you're interested in this kind of thing, they were able to actually do single cell RNA-seq on the cells from their fine needle biopsies. So there might be, so for those people who study that, there might be some interesting things there. And they had 24,000 cells, so quite a few more than in the last paper. But yeah, they just, overall their conclusions are you do get better germinal center response if you boost in the same lymph node. But for the overall antibody response, it all evens out in the wash after about 4 weeks.
So they have a study here, the discussion.
Yeah.
Is real-world data from a retrospective analysis of 2.6 million people.
Yep.
88% had ipsilateral boosting, and then 20 had contralateral.
Yep.
And short-term effectiveness was greater for the ipsilateral with less frequent PCR positivity. But I don't believe that's right because we're not preventing infection. We're preventing disease. So I don't know what.
We still don't have the study, basically. Yeah. So, like you said, if you're going to get exposed in the next week, if you boost in the same arm, you're probably better off. But if you're not sure, then it probably doesn't matter, according to this study.
But I think on top of that, the mechanistic insight from this is really considerable, right?
I thought so. That's why I thought it was a really good paper, because I think, I think it does matter that these subcapsular sinus macrophages mature in some way, which is probably due to some adjuvant effects, right? And some probably epigenetic changes that they change their secretion of cytokines or whatever that communicate with the B cells and change that microenvironment. So they really do change the lymph node. In the end, what those changes have in the overall outcome of disease may be less important.
Yeah. I would really like to know what Gabriel Victoria and Michelle Nussenzweig think about
this. Yeah.
Right?
I agree.
Because they're very interested in these things.
Yeah.
It's cool. Well, by the way, the pictures here of the kind of three-dimensional of the lymph nodes, you can see when the antigen's on the surface versus inside. It's very cool.
Yeah, it's really neat. Yeah. They have some videos too.
Nice.
Yeah.
All right.
That was fun.
Thank you. That is Immune 93. You can find the show notes at microbe.tv/immune. If you want to shoot us any questions, immune@microbe.tv. And if you enjoy these programs, we'd like to have your support. We give you real science, not lies, not misinformation, nothing politically motivated, just pure science. And these days, that's hard to find.
It is.
So if you like that, we'd love your support to continue. Microbe.tv/contribute. We depend on you. We don't do ads or any other source of income. We depend on donations. So microbe.tv/contribute. Cindy Leifer is at Cornell University in my old stomping grounds. Cindy Leifer on Blue Sky. Cindy Leifer on X, and Cynthia Leifer on Threads. Thank you, Cindy.
Thank you. This was fun.
I'm Vincent Racaniello. You can find me at microbe.tv. I'd like to thank nobody, actually. Nobody does the show notes. Nobody. No. I'd like to thank Tatami. That's where the music is from. Thanks for listening to Immune, the podcast that's infectious. We'll be back next month.